All right, here we are again, but my background is a little different because I am back at school and I'm gonna see you guys in like 45 minutes, but we're gonna try this flipped learning style. So I will work through the PowerPoint as I normally would when you're in class, just you're gonna listen to it at your own pace. And then that frees me up to come and help you with the work when you have a question. So feel free to pause and rewind and listen to the information over and over again. But here we go. So we're talking about chapter 7, and we are focusing this time on 7.2, which is all about the innate immune system. Just doing a quick recap. So this is the key knowledge that we want to work our way through at the moment. So we've already had a shot at dot point number 1. Hopefully the words in bold there in blue you can definitely tell me something about, so, and use confidently in a sentence to describe them. So what a non-self antigen is, self antigens and allergens. Then the second dot point is strongly focusing on those pathogens. So that summary table in 7.1 of the different kinds of pathogens and what to focus on, and having a lot of detail really about viruses in particular and bacteria. That's one that we've already looked at. And now we're looking at the second sort of half of this dot point. So from here on. So we want to know about what physical, chemical, and microbiological barriers are in animals and plants, and characteristics and roles of components of the innate immune system in response to antigen, including steps of the inflammatory response. So that dot point there, that third dot point, it's a really good one to pay close attention to. So this is one of those classic examples of in your textbook, there's a lot more information than you actually need. So really highlight this dot point, and I've tried to bold it to make it nice and obvious. There are five elements of the innate immune response that you need to know the details of. It's kind of nice to know that there are other elements as well. And maybe appreciate that some of the names of them, be able to recognize that there are other cells, but the details, what you actually need to know about, are these ones: macrophages, neutrophils, mast cells, dendritic cells, and complement proteins. So those 5, keep the specifics to those 5. And likewise, being able to talk about those elements with the specific scenario of the inflammatory response. There's a little bit of detail given about what a fever is and what's involved in a fever. But it is the inflammatory response that's in your key knowledge that you need to know the specific details for. Okay. Right, so I still like to talk about the immune system in terms of these 3 lines of defense. It is a really nice, neat way, and there's cute little analogies you can use to describe it. But it's a nice, neat way to talk about how the body defends itself. It's very military style. So the first line of defense and the second line of defense, these are the ones that we're focusing on today. They are both innate. Innate means that they are always present. They don't need to have encountered the pathogen before. They are detecting self and non-self. So always present, non-specific. So it's not that they are responding to a specific invader. It's just anything that is non-self or foreign needs to be kept out. So that's first and second line defenses. Third line defense is a whole nother story. That's the special forces. They are the ones that recognize particular wanted individuals, those pathogens that the body has encountered before and stores a memory of, and will be able to act fast against in the future. So third line defense will save that discussion until 7.3. When you go ahead and do the style for this part, it does talk about third line defense. So there's a couple of videos in there, I think, that will give you some information about the third line of defense. Don't worry too much about that information. Just treat it as some prior knowledge, I guess, before you do 7.3. The focus is really for this one on those innate levels of defense, the first and second line. And that's a nice summary table to include perhaps in your own chapter summary, which it is kind of broken down into those first, second, and third line defenses, and lists a few examples nice and clearly. All right, so I've already kind of detailed what innate means, but it's nice to have a definition for it. So the innate response is non-specific, always present in the organism, and does not need prior contact with the pathogen to be produced, and protects against a wide range of pathogens. It can be physical and chemical. When we talk about the first line defense, we'll give a bit more detail of those. And cellular. Cellular responses are our second line defenses. All right, the first line of defense: physical, chemical, and microbiological barriers. If you download this PowerPoint, you can appreciate that I've color-coded your key knowledge. So up in the key knowledge, these words appear, and I've colored them the same color that they appear on the slide, so you can see exactly where you're lining this up with the key knowledge. Okay, so physical and chemical barriers, sometimes the line is blurred between the two because there are some elements of your body that can be accepted as both a physical and a chemical barrier. So I'll talk about those ones when we get to it. But physical barriers, these are the things that are protecting your internal environment from your external environment. So anything that is present in your external environment, to gain entry to your body, has to go through a series of obstacles in order to get anywhere. So those physical barriers that are separating, the first one, the most obvious one, is your skin. So your skin is the biggest physical barrier that protects the internal environment of your body from the external environment of your body. But then you've got to think a little bit deeper, because you have a lot of places in your body where the external environment is sort of within your body. So by internal environment, we mean that it's actually crossed over into your circulatory system, into the actual inside of your body. So inside your ear canal, for instance, it's not just out here that is the external environment. Inside your ear canal is also an external environment. But we've got other structures in there in addition to the skin that will stop pathogens from crossing from that external environment to the internal environment inside your ear. So things like cilia, which are tiny hair structures that will line the surfaces of things like your nose and throat. So tiny hair-like structures, and they act as a physical barrier. They will filter, they will catch, sort of waving there, they will catch whatever debris and pathogens might be entering through those areas. They work hand in hand with mucus. So we're talking up your nose and all that snot that gets produced. It is a defense mechanism from your body. So the snot, the mucus, will grip onto the cilia, and they together are forming this sticky sheet that pathogens will stick to, and then hopefully your body can remove them through a sneeze. And then we've gotten rid of that potential invader. And I mean, think about every potential entry point for a pathogen, and this is being discussed so much at the moment, talking about how you can protect yourself from this virus that we have at the moment, coronavirus. All the ways that a virus like that could gain entry to your body. So you're told not to touch your eyes. That's because your tear ducts are an entry point to the internal environment of your body. So the eyes, what's there to protect pathogens from entering your eyes? You've got your eyelashes would be a physical barrier. Tears can also count as a physical barrier. So the action of those tears welling up and falling, removing whatever's caught in them, that is a physical barrier. Earwax inside your ears can be considered a physical barrier. Anything that is physically blocking a pathogen from entering that point of entry. Eyes, ears, nose, throat, even reproductive organs. And that's where the microbiological barriers come into it. So we have, and the female reproductive organs are a really good example of that. We've got a normal amount of, or culture, growth of bacteria and microorganisms, yeast, that will live in those passageways. And if they are living there, that means that other more harmful things can't take up residence there. So microbiological barriers work by being a healthy, normal, natural population of microorganisms that are blocking any other kind of pathogen from potentially taking up residence in that area. And the analogy could be, I guess, like a garden. So if you have lots of plants growing in a garden, there's less potential for weeds to grow in there because they're competing for that environment. So that is the analogy to use with microbiological barriers. Okay, so physical barriers, anything that is physically keeping a pathogen from entering any of those entry points that we've discussed. Chemical barriers, and I have discussed microorganisms, so we can leave that one as well. But chemical barriers are the list is a bit shorter, but it's some of the ones that I've already discussed for physical barriers, they cross over. So the ones that you want to go to talk about for chemical barriers, any kind of chemical substance within the body that has the potential to break down a pathogen. So that keyword breakdown is how we're distinguishing between the physical and the chemical barriers. So chemical barriers are things like enzymes. Lysozymes is a class of enzymes that actually do the job of breaking things down, and it's one of those good words in bio that you can work out what it means just by looking at breaking the word down. So zymes kind of sounds like enzymes, although we know that enzymes, the names often end in A, so it doesn't quite fit with that pattern. But lyse, hopefully we recognize that prefix, that term as breaking in biology. So this is a class of enzymes that breaks down cells. Lysozymes are present in things like tears, sweat, and saliva. And we've already at least talked about tears as being a physical barrier. Well, it turns out they contain these enzymes, which are it makes them a chemical barrier as well. Although if you're going to talk about tears as being a chemical barrier, then make sure you refer to the enzymes that are contained in them, these lysozymes. And then the other one to be able to mention would be stomach acids. So your external environment continues all the way down your esophagus and into your digestive system and all the way out the other end. It's all external environment. So we need lots of barriers in place to make sure that food, that things that you're intentionally taking into that environment won't be able to cross into your internal environment from that external environment that's contained within your digestive system. And a big one there is your stomach acids. So the acids that we find as part of your digestive system are working not only to break down your food into manageable sizes for absorption, it's also working to break down any potential hazardous microorganisms or virus, whatever it may be, any of those pathogens that might be entering your body. Not 100% effective, as we know. You can suffer things like food poisoning from taking that bacteria into your body, and it's obviously not, the stomach acids are not strong enough to get rid of all of it, but all of that kind of process is your defenses, your body defenses trying to get rid of that pathogen too. So it's all your body trying to do you a favor. But the stomach acid should be working, hopefully, to break down those pathogens before it gets too far. All right, and that leaves us to get into your second line defenses. So these are our cellular responses. Before we talk about the actual cells involved, the ones that you need to know about, there's a few molecules that we want to be familiar with and what they do as part of this second line of defense. All right, so the first one we're already familiar with from looking at signaling molecules. So cytokines, hopefully you remember that just in general, we talked about them being signaling molecules of the immune system. What do? They indicate damage or potentially dangerous invaders. So they are signals produced by cells, and we know that as part of apoptosis, they can trigger the death receptor, the extrinsic pathway of apoptosis. So they're doing a lot of things in the immune system to try and keep those pathogens from invading the body. Interferons are they are a cytokine, and cytokines is a very big group, but interferons are a specific one that gets mentioned, and their job we'll mention again when we talk about the third line of defense, but they act by virus infected cells, so a cell that's been infected by a virus, as part of the detection that it has been infected by the virus, they release these things called interferons, and interferons are a signaling molecule that will trigger the third line of defense to come and get rid of them because they recognize that they themselves are infected. And complement proteins, it might come back to this slide, but I'll talk about those ones in a little bit more detail. Can I just point out too that I've misspelled it here, which is probably a good teaching moment. Complement should be spelt this way with the E, not with the I. All right, so these cells of the innate immune response. Where can I put my face? I'll cover part of the heading. Okay, so the ones that you need to know about, and I've put them down here again in bold: macrophages, neutrophils, mast cells, dendritic cells, complement proteins. Oh my goodness, there are so many cells involved in the second line defenses. Even macrophages is a huge class of specific macrophages that do different jobs. So keep it to these 5 elements that you know the details for. Okay, so mast cells. What do we want to know about mast cells? Important things. Mast cells they're sort of like the first responders. They're the guard cell of the immune system. So they reside in peripheral tissues. So they'll sit in skin layers. They don't migrate. They really sit close to the periphery or close to the surface of any region where the external environment meets the internal environment. And what they do, so the most important job that a mast cell has is it detects disturbance. It recognizes when there has been an injury or potential invasion, and it releases a signaling molecule called histamine. So mast cells release histamine. That one's going to be really important soon, and I'll keep referring back to that one. But just for now, if you can file away, mast cells release histamine. Histamine has a really important job, so that's something that we'll come back to soon. And that's their job. So mast cells reside in peripheral tissue, release histamine. Macrophages. Macrophages move through the lymphatic system, so just through your lymphatic fluid. They can migrate from blood vessels and move through tissues. Their biggest role is that they can release cytokines, so they can signal each other, they can signal other cells, and they are a phagocyte. So phagocytes are cells that do exactly what this diagram is sort of showing here. They engulf and digest foreign material. It's not always a pathogen. It might be apoptotic bodies. But anything that needs to be cleaned up within your body's lymphatic system will be digested and broken down by a macrophage. So macrophages can release cytokines to signal other cells, and they are phagocytes. They engulf and digest foreign material. Dendritic cells, all right, things that we need for dendritic cells. Look at the shape of it. The shape is really important. They have that kind of star-like shape with projections hanging off the sides, and that kind of fits nicely with what we learned about neurons and the shape that they have. So remember, dendrites on neurons were those projections that come away from the cell body, those bits that hang away from the neuron cell body, and dendritic cells have that kind of similar shape. Definitely don't get them confused. So don't confuse your dendritic cells with your neurons. And it's got a very different shape all in all, but the word dendritic should trigger that kind of finger-like projection shape that it has there. Okay, dendritic cells. Okay, so they are the middleman. They are the runner, I guess. They are very similar to a macrophage. They do a similar job. They engulf and digest the foreign invader. But what they do, they digest it, and that's a little bit morbid. They reserve the antigen and they wear the antigen on the outside of their cell. So it's like they've taken a hostage, broken it down, retained its face, and they're now wearing its face on the outside of their cell. They then take that face, they take that antigen back to the third line defenses, and they say, hey, this is the bad guy. All right, what I'm doing right now and the way I'm describing it, I am really personifying these cells, which you can't do. You can't use that in an exam to describe the action of the dendrite, of the dendritic cell, but it is a nice way to remember it. So they are antigen presenting cells. That is the way you would describe it in an exam. So you take, or the dendritic cell takes the foreign invader, breaks it down, retains the antigen, and then presents that antigen to the third line defenses. And more detail will follow on that in the next chapter. Okay, so that's the big role of the dendritic cell. And then the last one, of the cells anyway, is the neutrophil. For your intents and purposes, neutrophils are very, very similar to macrophages, with the key distinction that although macrophages can travel in bloodstream, it's not something that VCAR really focuses on. So if you're told that there is a second line cellular cell that has been observed and it's migrated from the bloodstream, they're telling you that it's a neutrophil. That is the distinguishing characteristic to VCAR that they're talking about, a neutrophil. So neutrophils have a very short lifespan, and they do the same job as a macrophage, but much more aggressively. So they will come in, they migrate from the bloodstream, they come to the site of an infection, and they really go to town on the bacteria, on the invader. So they are a phagocyte. That's their job. They are taking in and breaking down the pathogen, but they're doing it at a much faster rate. So neutrophils migrate from blood vessels and are phagocytes. And then the last one out of that set of 5 that you've been asked to have details for are our complement proteins. So complement proteins, although not cells themselves, but the things that you want to know is that there are 20 kinds of complement proteins, and they do 3 things. They can cause bacterial cells to rupture, so they are effective against bacterial invasion. Some elements are effective against both viruses and bacteria, and some are only effective against one or the other. This is one that will only work on a bacterial invasion. So they can cause bacteria cells to rupture. They stick to bacterial cells and highlight it to phagocytes, so they act as a marker. They will attach themselves to the surface of a non-self cell, and that effectively tags it as being something that is foreign and needs to be gotten rid of by phagocytes. And promote inflammation, which brings us then to, and that is kind of a graphic of what complement proteins would do, so punching holes effectively in bacterial walls. Just a cute little graphic for what phagocytosis is. Remember the word. And that brings us to inflammation, so the body's inflammatory response. Okay, so when external barriers are breached, inflammation is switched on by damaged tissues or invading pathogens. What are the steps of inflammation is something that you're being asked to walk me through this week. The best diagram to represent what's going on with inflammation is this one out of your textbook. It's a very nice summary of the story of inflammation, and it involves nearly all of the players that we've just gone through. The only one missing from it, which you would need to add in the detail yourself, is the dendritic cell. All right, but the story goes something like this, and you can definitely have a read through all of the nitty-gritty details that are displayed here. But I want you to be able to summarize it in your own words, and the story goes something like, when there is a disturbance, an injury caused, and a bacterial invasion takes place, the first responder is the mast cell. So the first step in inflammation is always that a pathogen has gained access to the internal environment of the body. That is an infection, by the way. That is the only definition of infection that there is, that a pathogen has been detected in the internal environment of the body. So an infection has taken place. Mast cells will detect that there is a foreign invader. They release histamines. Histamines have a few jobs. So histamine, the probably three biggest roles of histamine that you can file away. Histamine, well, in general, it triggers the inflammatory response, but what that actually means is it's increased blood flow. So lots more blood is coming to the area. It increases vasodilation, which means that there's a lot more potential for cells to move in and out of those blood vessels. So increased blood flow, increased vasodilation, and it is a signaling molecule itself. So it is triggering more second-line defense cells to come to the area. Kind of like ants leave that chemical trail for other ants to find a food source. These histamines attract cells to the area of infection. Okay, so our mast cells have released their histamine. From the bloodstream now, we have neutrophils migrate. Neutrophils are phagocytes and will begin digesting these bacterial invaders. As part of what the histamine's role is, it's also attracting macrophages to the site of infection. So both macrophages and neutrophils are working to digest and get rid of that bacteria. The macrophages are releasing cytokines, which is in turn encouraging more and more second-line defenses to come to this site of infection. So this is effectively what pus is. When you see that response happen at the site of an infection, that is your white blood cells gathering and attracting more and more white blood cells to that site to get rid of the bacterial invader. All right, what's missing from this diagram is the role of that dendritic cell. So you would add in your dendritic cell here, make sure that you've got that nice dendritic shape to it, but it's doing what the macrophage is really doing, but with the added element of that antigen presenting. So breaking down the bacteria and then wearing the antigen on its surface. And that is done using MHC class 2 antigens. So wearing that antigen then on its surface, and it will migrate then back to, through the lymphatic system, not through the bloodstream, through the lymphatic system, it will migrate back to a lymph node. And that's where I'll end the story of the dendritic cell, but that's where we'll pick it up from when we come to talk about the third line of defense and what happens next after that point. Okay, so that brings me to the end of the content for 7.2. Go back and have a look and watch it again and do what you will with it. But that's the theory, and I'll be around to help you with the questions and the work.